It's my pleasure to speak to you tonight. What I'm presenting here is something we prepared within the EUPATI IMI project. EUPATI stands for European Patients Academy on Therapeutic Innovation, a project that has ended in January this year. We developed material for patient organizations to learn about medicines development. And in this webinar we prepared here, we wanted to show people how actually patient organizations can work with industry and make something unthinkable happen. And we have two examples in this presentation, one where we went to industry and the other one, actually the same company where they came to us to get advice on something. I also admit to have been part of the CIOMS guidelines working group that Ingrid nicely quoted before. So we start in HIV drug development. HIV drug development is a success story, but it was a bumpy road. And we found ourselves in the around 2004, 2005, in a picture of huge problems due to accumulated drug resistance. Many patients had already lost their life back then. And in 2003, a treatment from Basel was approved from Roche, T20. This was more or less the last resort of people. It was hugely expensive. It had to be administered via syringe twice a day in the back of people. And it was really a pain. People required a nurse to come to see them twice a day. So everybody was really desperate. And multi-drug resistance was considered to be the big problem of HIV treatment back then. We had this picture in the Swiss HIV Cohort Study with patients who started treatment before 1999. 56% had drug resistance mutations, 22% had 2-class resistance, 11% had even 3-class resistance. That was the picture worldwide back then. We lobbied for overcoming this drug resistance, and we knew how people had accumulated this resistance over time by going from one trial to the next, always adding a new compound, and always developing again resistance to the new compound in the regimen. So we thought that we could only get rid of this if we would have a pivotal trial combining two investigational compounds in one study. But we couldn't find the two companies who would put their two compounds in one study. But then we found one company that had two drugs in two classes, and they were ready to put them into one study, and EMA and FDA agreed that this was a sensible idea. So now the company slides, they really helped us a lot making this happening. Never before two investigational compounds were put together in one pivotal phase 3 study. The compounds, the darunavir, Prezista, TMC-114 back then, was a protease inhibitor, and TMC-125, etravirine, was a non-nucleoside analoga. Both have demonstrated to be effective against resistant drug strains because Tibotec had this knowledge in-house. They were also doing resistance testing in a sister company. So that was their situation. They got advice from externally that this was actually a good idea. Everybody was convinced this was the thing to do. However, they had internal resistance to overcome because this was not a no-risk strategy for them. And in the end, they decided to follow what was proposed, what everybody expected them to do, and they run out the trial. So there was a risk of a negative phase 3 study. That's one thing. There was a potential that one drug would influence the other one on contraindications and on precautions, because we didn't know the two compounds well enough. and there was also the risk that a bad outcome could influence the label of the other one. Here you see the phase 3 design of the duet trials. We have one arm with TMC-125, that was the very new compound, the NNRTI, and the background regimen. In the second arm, we had a placebo for 125 and the background regimen. And the background regimen was the protease inhibitor boosted with ritonavir and optimized NRTIs. Optionally, enfuvirtide T20 was also allowed because we were in a heavily pretreated population. So that was the design. We had 2 duet studies. Recruiting, duet 1 had only people from France and Europe, otherwise it was global. Duet 2 was mainly a European study, with also some US and Australian participation. And here you see the results. After 24 weeks, everybody was very, very nervous when this study was running. And already in 24 weeks, you see that the TMC-125 arm was doing far better than the placebo plus background regimen arm, with only one new compound in the arm. And you see in the stratification that this actually worked at, this was very marked, of course, when no effective drug was in the second arm, but it also played out when one effective drug was in the second arm, and it still made a difference when more than 2 effective drugs were in the second arm. So really a successful outcome that exceeded our expectations. The FDA label was good, didn't have any implications. The EMA label, however, didn't allow the use of TMC-125 Intalens without the protease inhibitor in the beginning. And this is something that has had a negative impact on the introduction of the second drug in the European market. So something that probably today wouldn't happen again, but 10 years ago it happened. Lessons learned for the company. That it was good to involve patients early on and to listen to them, and that it was also a very good thing to involve both the patients and the researchers in the same advisory boards and have them speaking together and combine what they both said. We had patients from the US and Europe reviewing the protocols. We also met together because we were talking both to the EMA and to the FDA, and we needed to meet and to see us and to know us in order to tell both agencies the same thing. And there was also a patient on the DSMB while these studies were running. And the environment today in HIV is that these duet trials have actually formed what is standard today, that the concept of individually optimized HIV treatment regimens to maximize the number of fully effective agents is pretty much standard of care. And we could actually learn something from this experience in hepatitis C. And as a result, drug resistance in the HIV cohort study, here you see it clearly, people who have started treatment after 2007, only 10% have resistance mutations versus 56%, and 1.7% have 2-class resistance versus 22%, and 0.2% have 3-class resistance, that's almost nobody, versus 11%. So drug resistance, at least in Western Europe and North America, is history since that trial. Now we come to another example with a different compound, more your field, because it's a nanosuspension. Long-acting rilpivirine was developed from about 2007, and this drug had the potential to improve adherence and convenience in people living with HIV. So in the mid-2000s, treatments were moving to less frequent dosing. We had first single tablet regimens, but we also knew that people could get tired over time with twice daily dosing and the requirement that 95% of the drugs had to be taken. So adherence levels lifelong are still a challenge in HIV. And I tried to speed up. Janssen had expertise in making nanosuspensions that could make a long-acting injectable formulation reality once. And they wanted to hear from us if we were interested. And this is the drug, long-acting TMC 278, a depot formulation given intramuscular that could potentially lead to a once-monthly injectable drug cocktail. And that could also be used in maintenance, and that could be used in prophylaxis of HIV infection. And it would also help people to have to be more adherent, to go to holidays without drugs, etc. So they called advisory meetings in December 10, 2007. I participated in this one, where we all met, the researchers, scientists, and us from the US and Europe together in Antwerp, and we discussed these plans. Our biggest concerns from the patient side were resistance, still a big issue back then, long-term adherence. We knew how challenging this was. GI side effects was another issue we had. The physicians were not with us on that one, but we felt it every day, that this was a pain, and we thought that an intramuscular suspension could help us overcoming this problem too. And we knew that ART, antiretroviral treatment, could work as prevention, but we didn't really know how back then. And despite the lively memory still back then of injected drugs, remember T20, we thought if the injection would happen only once a month, it would be something we could accept and live with. And once a month versus twice daily, that's a different world. And we also saw that use in prevention could be very important, especially thinking of people living in the south in Africa, that the once monthly injection could protect them much better. But we had the concern that an oral lead-in had to be available because this nanosuspension cocktail would remain in the body for a long time once given. So here you see the study design, the latte study design. These studies are ongoing at the moment. They are combining, they are trying to put an entire drug cocktail into this nanosuspension. It's currently an integrase inhibitor which is being tested, GSK 744 it is called, it still has no name, and the studies are ongoing. A year ago, Janssen signed an agreement with Healthcare, which is a young company coming out of GSK, to work on this drug development plan. And in 2017, when we now look back on 10 years of discussing this potential treatment, the Latte 2 32 weeks results were presented a year ago. Other studies are ongoing. In summer 2016, I had reviewed 2 phase 3 protocols on that. The assumptions we made in 2007 still hold true. It was still, everything was valid. The combination with the integrase inhibitor that happened later, we didn't know 10 years ago with what they would combine the drug, turns out to be a very successful coincidence because the integrase inhibitor is today considered to be the most powerful and the safest drug we have in the HIV treatment armamentarium. And drug development is a long road. I'm personally looking forward to a drug holiday, to a holiday without having to carry pills with me. So I'm quite excited about this to come along. And PrEP, in the meantime, pre-exposure prophylaxis, has become a successful prevention intervention, especially for gay men in the United States, in France. Scotland has recently approved the use of PrEP. Switzerland is very resistant, but I'm hopeful still. And other countries will follow. Israel has also approved the use of PrEP. The conclusions is that early collaboration, including patients, is possible and productive. Prerequisites, top management in the companies has to support that. And there need to be established links between the company and the trained patient group. And there need to be established links between the patient group and the academic researchers and the regulators. Despite all this being, all these requirements being fulfilled back then in 10 years ago by my organization, we still had the problem with the label in Europe when Intalens came on the market. So not everything played out fine. And I would like to thank Brian Woodfall from Janssen, as it is called today. Janssen bought up Tibotec for having helped me with the slides and contributed back then in 2007 to make all this happening. Thank you.